Guys, welcome to Danso's online biology for easy mastering of your biological problems. And today we're going to look at some of the issues that has to do with gene mutation. Now, mutation is going to be our main focus today, and we shall look at mutation in so many respective patterns. Now, mutation, we know it has to occur in our genes. A gene, which is a segment of DNA that code for a specific protein to mastermind the phenotypes that we possess. And when you look at mutation, it has to occur in the DNA structure. And when you look at the DNA structure, there are nucleotide bases, and these bases will be changed. We have nucleotide bases like the guanine, the cytosine, the thymine, and then the adenine. So when we said mutation, these structures would be changed because normally from the Garkov principle in gene, the nucleotides will match. They will align in a very specific manner. That is, G and C will always align, and T and A will always align. So they will bond. Thymine and adenine will be bonded by a hydrogen bond, and guanine and cytosine will also bond by a hydrogen bond. But when mutation comes, they can change this cytosine to adenine so that guanine and adenine will bond. That is a substitution. So it is a mutation. Mutation by definition is any changes that occur in the nucleotide sequence of a gene leading to some changes in the phenotypes of an organism, or leading to some malfunction of that particular gene region of that mutation. And to begin now, we have a rearrangement of chromosome that is also considered as a mutation. A chromosome will rearranged. It can be in a form of substitution or transition or transversion. Now, this mutation occurs spontaneously. It occurs spontaneously, and sometimes it can be a very fast rate or it can be a slow rate. That all depends on what we call the mutagenic. Mutagenics are those chemical substances that can accelerate the rate of mutation in the systems. So mutation has a direct link to those chemical substances, like smoking cigarettes, exposure to the light. All those things are mutagens. They accelerate or increase the rate of mutation, that is the change of these nucleotide sequences. So we also understand that, like as I said, if you are exposed to too high a level of mutagen, that is those chemicals, they can threaten the survival of a species. Those chemicals, because when mutation happens, some mutations are very detrimental to the survival of a species or a particular organism. So we have seen this case study of a frog species that are found in America, Washington, and Oregon. So these frog species, they faced a very high extinction rate. And the research shows that the main reason for that is because of the atmospheric ozone layer has been reduced a lot, and that results to an increase of ultraviolet radiation. The ozone layer was destroyed, and in other words, the sunlight was coming in a very intensive way, direct contact with these frogs, and led to these frogs being extincted. Probably they cannot handle that extinction rate. So that's one of the reasons why mutation is dangerous. So to sum up everything here, mutation is a change of gene sequence or nucleotide sequences. Either the guanine or thymine will bond, or adenine and thymine will bond. We call them the purines and then the pyrimidine. So they can be all this kind. When this happens, it leads to a very uncomfortable condition. Sometimes the gene region where it's affected, that gene region becomes silent. So thank you for watching, and in our next lecture we will look at the main types of mutations and get to you. Thank you.